Hello gang, and welcome to Unit 19, where we're going to talk about the immunoassays. We did try to get immunochemistry and serology and immunology a little bit in microbiology, so stay tuned while we discuss ELISA and immunofluorescence. ELISA is an acronym for enzyme-linked immunosorbent assay. Here, an antigen and antibody can be labeled with an enzyme, and there are two different types of method, the direct ELISA method and the indirect ELISA method. In the direct ELISA method, antibody and conjugate are on cellulose acetate paper, and they migrate across the cellulose acetate paper to give you a result. In an indirect ELISA test, there's antigen in wells, and we incubate the plate with substrate and then add peroxidase. If antibody is present, the substrate color will change. Okay. Indirect ELISA is used to detect infection by testing patient's serum for the presence or absence of antibodies against a pathogen. So in other words, if our patient's antibody titer goes up, it means that if it's very high, they probably have an acute infection due to that particular pathogen. In an indirect ELISA, a plastic plate is covered with antigen. This is also known as solid phase technology, and the antigen reacts with the antibody in patient's serum. So in indirect ELISA, we have a 96 well plate, and that plate is coated with antigen. Okay, if our patient has antibody in their serum, once we add their serum to the antigen in the plastic wells, they're going to link up, and we're going to add an enzyme labeled antibody conjugate. If antibody is present, then the conjugate reacts, and the conjugate is basically a color enhancer, and the enzyme activity is measured spectrophotometrically after the addition of chromogen. Okay, so spectrophotometrically, here we're looking for a measurement of light relative to wavelength. The enzyme either absorbs or transmits light at a certain wavelength. There's two approaches to see if our patient has a disease to a pathogen. We can actually test for specific antigens from that pathogen, or we can test for a specific antibody that is specific to that antigen. ELISA, again, is enzyme-linked immunosorbent assay, and it's a method that we can use to determine if we have specific antibodies to a disease or if we have antigens to a bacterial pathogen or viral pathogen. Okay, this method, or the methods performed in a 96 well plate, are known as indirect ELISA methods. ELISA is usually reported using some sort of numerical result in a calculation from the optical density. Enzymes used in the methodology help detect or quantitate immunologic reactions. Here we have enzymes that are linked up with conjugates, giving us a visual clue as to the reaction strength. So in other words, if our 96 well plate has a very strong yellow color, it means that our antibody titer or our specific bacterial pathogen antigens are very, very increased. Okay, enzymes used in this assay are horseradish peroxidase, alkaline phosphatase, G6PD, which is glucose 6-phosphate dehydrogenase, or beta-galactosidase. We can actually use indirect ELISA to detect lots of diseases, including measles, mumps, HIV, hepatitis B virus, West Nile virus, avian influenza, etc. And there are actually three different types of antibody detection, three different types of ELISA procedures. One is called non-competitive, one is called competitive, and one is called capture. In non-competitive indirect ELISA, we use excess label specific antibody towards an analyte of intent. In competitive ELISA, we use a labeled analyte, and we look for a concentration of antigen. We have labeled antigen and sample antigen that compete for a binding of antibody. And in capture ELISA, we use capture antibodies specific to a target antigen. This is also known as sandwich ELISA. To determine if our wells are positive in an indirect ELISA, what we do is compare it to the blank well. Usually our first well is the blank well, and we actually can minus out the blank from our concentrations of optical density. So if you minus out that blank, so for example, if you have a blank optical density that is 0.005, we can run two standards, and we're going to take the average of those two standards. Okay, then what we can do is take the average of those two standards and minus out the optical density value of the blank, and then we're going to actually divide by the mean of the two standards. Okay, this determines if our wells are positive or negative for the antibody. Anytime you're doing an ELISA procedure, you always want to pay close attention to the package insert on how to interpret optical densities as either being positive, negative, or equivocal. Equivocal basically means that our result is in the gray zone and that we can't really determine if the well is positive or negative. Many kits use a value of 0.9 for the optical density and less to be considered negative. An optical density on most kits of 0.91 to 1.1 is considered equivocal, and an optical density of greater than 1.1 is considered to be positive. Depending on if capture ELISA is used to detect IgG or IgM specific antibodies, that's going to determine whether or not the disease is in the acute stage or the convalescent stage of the disease. If you're in an acute stage of the disease, usually it's your IgM specific antibodies that are elevated, and then your IgG specific antibodies are going to increase when you are in the convalescent stage of the disease, or if you have a successful vaccination. If you have a successful vaccination, your titer is going to be much lower than in the case if you have an acute disease. All right, hopefully this will work for you. If you click on the link, you should be able to see a little video about the ELISA procedure. If it doesn't work, I'll just go ahead and put the video in as another link. ELISA is a nice immunoassay for detecting infectious diseases where they're too difficult to detect in microbiology, whether that be a viral disease or a disease of a fastidious organism that's too difficult or slow to grow in microbiological media. So the ELISA immunoassay can look for lots of infectious diseases, including the Lyme disease spirochete, which is Borrelia burgdorferi. We can also look to see if our patients are positive for cytomegalovirus or negative. We can look for antibodies against hepatitis A and hepatitis B. We can look for antibodies against HIV, human B lymphotropic virus, Shiga toxins, which are the toxins associated with E. coli 0157H7, Clostridioides difficile, West Nile virus, rubella, measles, mumps, and toxoplasmosis. Those are all either viral diseases or diseases that are tough to identify through microbiological means. So this is what a plate looks like for an indirect ELISA test. And you can see we have 96 plastic wells that fit into a well holder. Okay. In our first row, we can see that our first well up there in position A1 is our blank well. Okay, then we add our two calibrators. Usually we run those in duplicate. Then we have our negative control, our positive control, and then our patients follow that. Okay, so basically what we do is we run our sample after we go through all of our substrate and conjugate phases, and we hit the stop phase, then we run this on a spectrophotometer. Okay, the spectrophotometer is going to give you an optical density for the blank, the calibrators, the negative control, the positive control, and all of your patient samples. So what we're going to do is take the patient and control values and minus out the blank. That gives us an actual value that can actually be divided by the mean of the two calibrators, and that determines whether or not our wells are positive or negative for that antibody. In immunofluorescence technology, we're looking for antibodies that are labeled with a fluorochrome. A fluorochrome is a chemical that can absorb light energy and at the same time emit light at a longer wavelength. A fluorochrome that is chosen is usually one that can absorb UV in short blue light and emit light in the visible range. The fluorochrome is either activated or excited by the particular wavelengths of light. The fluorochrome that is needed in immunofluorescence technology in most laboratories is fluorescein isothiocyanate, which is also known as FITC. This fluorochrome is actually blue and it allows for maximum absorption, so emission is in the yellow-green range. So what we're looking for are yellow and green inclusions on our slide, whether that be yellow or green giardia or cryptosporidium, or else we're looking for yellow-green cells of RSV. But we're always looking for a yellow-green color whenever we use fluorescein isothiocyanate. We have two different types of immunofluorescence procedures. One is called the direct fluorescent antibody procedure, and one is called the indirect fluorescent antibody procedure. The direct fluorescent antibody procedure is used for identification of such diseases as RSV and Giardia and Cryptosporidium as far as our parasitic diseases are concerned. In the direct immunofluorescence test, we have the antigen, which is the unknown element, plus our specific conjugate, and the antibody is the known element. And we're primarily used to detect nucleic acids in organisms. Okay, so in the direct fluorescent antibody test, we take the patient's sample, we put it on a slide, we add our FITC, and then we allow it to incubate. After incubation, we wash the slide, and then we observe it using a fluorescent microscope. Anything that fluoresces green indicates a positive test for whatever analyte we're looking for. In indirect fluorescent antibody testing, we have an antigen in patient serum that is incubated along with the fluorescein isothiocyanate labeled anti-human gamma globulin. This is the conjugate or the specific antibody that's attached to the fluorochrome. We incubate again and observe for fluorescence or no fluorescence. Antibodies that act antigenically and react with anti-immunoglobulins can be observed using a fluorescent microscope. Okay, circulating patient immunoglobulins can react with the nucleus or nuclear components of the host tissue. We saw this when we were talking about anti-nuclear antibodies in the immunology course. So for looking for anti-human antibodies, anti-nuclear antibodies, what we can do is take a donor cell, which happens to be a human epithelial cell, and we can look at that nucleus and how it fluoresces. If the nucleus fluoresces a certain way, it may indicate that they have lupus, or it may indicate that they have HIV, rubella, or even toxoplasmosis gondii. Here we can compare the direct fluorescent antibody example to the indirect fluorescent antibody sample. You can see in the direct antibody sample, we have the cell that is a donor cell, and it is attached with antigen, okay? And then the antibody and the fluorochrome link up to that antigen, causing fluorescence. In the indirect ELISA test, here we have the cell, which is a donor cell, and antigen. We have the antibody that's attached to the antigen, and then we have secondary labeled antibodies with FITC. There are some advantages and disadvantages to fluorescent antibody testing. Some of the advantages for using fluorescent antibody testing is that it's rapid, okay, and that it's very specific. Okay, so an advantage of fluorescent antibody testing is that we can rapidly identify the antibody being present in cases such as Bordetella pertussis, or RSV, or Giardia cryptosporidium. It may actually eliminate the need for culture because it is so sensitive and quick. A pure culture is not necessary. You can use a mixed culture and you're still going to only get specific fluorescence. And also it's very sensitive, so only a small number of organisms is required to get the sample to fluoresce, and the procedure is quite simple. Some disadvantages of fluorescent antibody testing, for one, it's very expensive, and personnel must have a great deal of expertise in fluorescent technology and fluorescent microscopy in order to view these samples. The fluorescent microscope has several parts that I want to talk about. It has a condenser very much like a bright field condenser. We have our oculars and objectives that are very much like a bright field microscope. And then we have a couple things that are unique to a fluorescent microscope. For example, we have a heat filter, which is located between the light source and the exciter filter. And this actually removes heat energy from the light source and protects the other filters. The exciter is located between the heat filter and the specimen, and it stops visible light from passing through the specimen, but it allows UV light to pass and transmits the wavelength of light compatible with the fluorochrome. So the fluorochrome is actually going to absorb this light. Then in the fluorescent microscope, finally, we have the barrier. The barrier is located between the specimen and the observer, and it transmits wavelength emitted by the specimen and retards light above 500 nanometers. And then we have our light source. Okay, there's actually two types of fluorescent microscopes, including the transmitted light microscope and the incident light microscope. So we have two types of fluorescent microscopes, including the transmitted light microscope and the incident light microscope. Okay. In the transmitted light microscope for fluorescent microscopy, light is passed through the specimen and is partially absorbed, and we use a dark field condenser, and the specimens are viewed on oil. In incident light, we have light that is focused on the surface of the specimen. None is actually absorbed. There's no condenser needed, as the objectives also function as the condenser. The light beam splitter reflects shorter wavelengths down through the objective and transmits only longer wavelengths back to the ocular. In our laboratory, we do use an incident light fluorescent microscope. Here we have the light, and next to the light we have the heat filter and then the exciter filter. Then in front of the microscope we have the oculars, and then below that we have a beam splitter that is built in with suppression filters and beam splitting mirrors. Below the beam splitter we have the objective, and then finally on the stage we have the specimen. So here in this drawing you can see that we have a light. Number one is the light source, and it passes through a density filter. And then 2, 3, and 4, we have fields that stops for color illumination. That would be the heat filter and the exciter filter that actually stops that for color illumination. We have the exciter filter and then the beam splitter, the objectives, and then the fluorescence emission from the specimen. Okay, we have the emission filter, which is up there above the objective, the infinite corrective objective, which is actually above the exciter filter, and then finally we have the binocular oculars. And then here is just a drawing of a reflected light microscope that is a fluorescent microscope. You can see it's a little bit different because it has that tungsten halogen lamp house, and a lot of our bright field microscopes don't have that. There's a lot of emerging fluorescent labeling technologies as well, including quantum dots, also known as Q dots. I'm not super familiar with this type of fluorescent labeling technique, but Q dots are used to label live biological material in vitro for research. Okay. We also have superconducting quantum interference devices, also known as SQUID, which is used to measure extremely subtle magnetic fields and used to measure cardiac risk in the laboratory. We also have luminescent oxygen channeling immunoassays, and then finally we have FISH, or fluorescence in situ hybridization. FISH actually optimizes spectral features of filters to maximize contrast and brightness. It uses highly specific DNA probes to detect genetic markers with fluorescent molecules tagged with fluorescent antibodies. So that's a little bit about the emerging fluorescent labeling techniques: quantum dots, SQUID, Loki, and FISH.